Welcome back to The Explainer. Today, we're diving deep into the science and the ongoing debates surrounding alternative and repurposed treatments for cancer. We are going way beyond the conventional boundaries of oncology here, looking at how researchers and clinicians are actually trying to hack the very metabolism of cancer cells. Think about this for a second. Can existing everyday compounds unlock new ways to fight cancer? It's a hugely provocative question. Today, we're impartially laying out the clinical studies, the historical data, and the biological mechanisms to see what the source material really reveals. We're not here to take sides or push a specific agenda. Our goal is strictly to decode the science and understand what's actually happening down at the cellular level. So here's our roadmap for today. We'll cover 1, targeting the supply lines, 2, the vitamin C debate, 3, vitamin D and cell defense, 4, the folate paradox, 5, repurposing existing drugs, and finally, 6, the future of oncology. Let's start with section 1, targeting the supply lines. To defeat an enemy, you really have to know how it sustains itself, right? Well, solid tumor survival is entirely dependent on hijacking the body's blood supply through a process called angiogenesis. Basically, tumors are building their very own supply lines to siphon off blood, oxygen, and nutrients. And since just one single microvessel can feed thousands of tumor cells, targeting these supply lines is a massive, absolutely crucial strategy in modern oncology. This brings us to a second major biological hurdle, which is apoptosis. Normal healthy cells have a programmed self-destruct sequence that kicks in when they get damaged. But cancer cells, they essentially forget how to die. They just ignore these apoptosis signals and keep dividing. As we'll see, a lot of the therapies we're exploring today are designed specifically to force the cancer cell to remember that self-destruct code. Moving on to section 2, the vitamin C debate. Keeping those foundational mechanisms in mind, let's unpack one of the most hotly debated nutritional interventions out there, high-dose vitamin C. Historically, this sparked intense interest. The data from the famous Cameron and Pauling study reported significantly increased survival times for terminally ill cancer patients treated with ascorbate, which is just vitamin C. Their findings suggested these patients live substantially longer than matched controls across a bunch of different primary tumor types, and this literally spread like wildfire, putting vitamin C on the map as a potential anti-cancer agent. But, and it's a big but, the clinical data is actually full of stark contradictions. The Mayo Clinic tried to replicate Cameron and Pauling's results with rigorous, double-blind, placebo-controlled trials. The first Mayo Clinic trial found absolutely no significant difference in survival time versus a placebo. Their second trial found survival times were completely comparable to untreated groups. It's a perfect example of just how messy and conflicting medical research can be. However, more recent research suggests we might have been looking at it wrong. When given intravenously, like in massive pharmacological doses, vitamin C might actually have potential synergistic effects with conventional therapies by acting as a pro-oxidant that damages tumor DNA. Section 3, vitamin D and cell defense. Let's shift gears to a different vitamin. With vitamin D, the clinical data paints a much clearer picture of cellular defense. The anti-cancer activity here is tied to vitamin D's active form, which operates on a really profound cellular level by directly entering the nucleus. Once it's in there, it literally hits the brakes on the cell cycle, repressing the exact genes that allow cancer cells to multiply. Not only does it promote that crucial self-destruct apoptosis we talked about, but it also inhibits tumor angiogenesis. It's choking off the tumor's blood supply at the genetic level. This biological mechanism was actually put to the test in the Sunshine trial. Researchers compared high-dose vitamin D3, given alongside chemotherapy, against a standard dose for patients with metastatic colorectal cancer. The high-dose group saw a statistically significant improvement in progression-free survival. Meanwhile, the patients taking the standard 400 IU per day, they remained fundamentally vitamin D deficient even when the treatment ended. It really goes to show how crucial proper therapeutic dosing is when you're trying to influence cancer metabolism. Which brings us to section 4, the folate paradox. Okay, what happens when a vitamin that normally prevents cancer actually starts fueling it? This is the folate paradox, and it's fascinating. The critical factor with folate, or vitamin B9, is timing. Before a tumor ever forms, folate is your body's best friend. It's essential for synthesizing and repairing damaged DNA, which actively prevents cancer from starting. But once a neoplasm forms, the whole script flips. The cancer hijacks the folate, using it to rapidly replicate its own DNA and multiply. It literally becomes a growth factor for the tumor. And this paradox has some profound real-world implications. Certain chemotherapy drugs, called antimetabolites like methotrexate,
actually work by intentionally starving the cancer cells of folate. They block the cells from using it, which shuts down their DNA synthesis completely. This also explains why some observational studies found that excessive folate supplementation could actually increase the risk of advanced cancers. It's a stark reminder that in oncology, context is absolutely everything.
Now, for Section 5, repurposing existing drugs.
This whole idea of starving the cancer cell or reprogramming its broken metabolism brings us to one of the most exciting areas of modern research, repurposing existing drugs. Take the compound DCA, or dichloroacetate. Cancer cells typically survive by fermenting glucose, which is a highly inefficient process known as the Warburg effect. DCA tries to reverse this by inhibiting an enzyme called PDK. This stops the cancer cell from fermenting sugar and forces it to wake up its mitochondria and use normal oxygen-based energy production again. And here's a really vital piece of this puzzle. Thiamine, or vitamin B1, is heavily highlighted in the clinical studies as a crucial cancer treatment adjunct here. Why? Because thiamine acts as a necessary cofactor for the very pyruvate enzyme complex that DCA is trying to activate. You need it to make the mechanism work, which is why clinical studies routinely recommend thiamine alongside DCA to fully successfully reprogram the cancer cell's metabolism. It's not just metabolic drugs either. The sources extensively cover the off-label use of common antiparasitics in oncology. Look at ivermectin. In preclinical models, it inhibits tumor proliferation and metastasis by targeting specific genetic pathways. Then you have mebendazole, which acts almost like a traditional chemotherapy. It disrupts microtubules, which are the structural proteins a cell absolutely needs to divide, thereby triggering apoptosis. But the absolute kicker here is their combined target. Both of these drugs have shown an ability to selectively target cancer stem cells. Those are the very cells responsible for therapy resistance and tumors coming back. To see how this plays out outside the lab, researchers tracked a diverse cohort of 197 cancer patients who were prescribed a compounded combination of ivermectin and mebendazole through a U.S. telemedicine platform. This wasn't some perfectly uniform group. It was highly varied. Prostate and breast cancers were the most common, and over a third of these patients reported their disease was actively spreading at baseline. Now, here is the number that really jumps off the page from the follow-up data: 84.4%. This prospective observational study reported an 84.4% clinical benefit ratio at the 6-month mark. What does that actually mean for the patients? It means that out of the people who completed the follow-up, roughly 84 out of 100 self-reported that they had no evidence of disease, that their tumors had visibly regressed, or that their disease had completely stabilized. Now, we do have to pump the brakes for a second and look at the study design critically. The researchers themselves are very careful to point out that these are self-reported outcomes from an observational cohort. It is not a controlled clinical environment. While those high tolerability rates and self-reported benefits are undeniably compelling, they emphasize that these findings are hypothesis-generating. They are calling for urgent, rigorous, randomized controlled trials to truly confirm if these effects hold up under strict scrutiny. Finally, Section 6, the future of oncology. As we step back and look at the sheer volume of data across all these diverse interventions, from vitamins like thiamine and vitamin D to repurposed medications, what's the big takeaway here? The science of starving cancer supply lines and reprogramming its damaged cellular factories is relentless. It is evolving so fast. We've seen how timing completely alters the nature of folate and how simple antiparasitics might reshape tumor metabolism. It leaves us with a truly profound question. How will standard medicine and these repurposed therapies eventually integrate to conquer cancer in the years to come? Could it be that the next great breakthrough in oncology is already sitting quietly on a pharmacy shelf? I hope this explainer has sparked your curiosity to keep digging into the incredible science of human biology. Until next time.